So now we'll go to the last talk of this evening's first session, and we're going earlier down the evolutionary tree, so to speak, to karyorelictids, and Ying Yan is going to talk about preliminary analysis of genome architecture of Loxodes amongst the karyorelictids.
So, hi, thank you for giving me this opportunity to present here. And I will start with the conclusion and we'll go into detail later. So my hypothesis, I want to test, is protein coding genes in para-diploid Karyorelictea have low and equal copy numbers as compared to small subunit ribosomal gene. However, our preliminary results from Loxodes single-cell qPCR of 2 cells suggest that there are thousands more copies of actin than ELF1 alpha in the macronucleus. And also, as expected, SSU or DNA gene is highly amplified. So, first, I will give a really brief introduction of Karyorelictea, which we have great interest in. And this is a really diverse group of ciliates and have many different kinds of nuclei, which is showing here in this figure. Yeah. And most of them are marine. The only exception is the genus Loxodes, which are highlighted in red box here. They live in gross freshwater. And to talk about the phylogenetic position, Karyorelictea is sister to Heterotrichia and really far away from the well-studied ciliates in Oligohymenophoria, Spirotrichia, and Phylophryngia. So this is a phylogeny of ciliates, and the well-studied ciliates over here, these large branches, and our focus is down here. Actually, karyorelictids are pretty difficult to work with. One reason is that they are largely uncultivable. Only Loxodes has been reported to be cultured, but sadly we haven't got success with that. And another main character which makes karyorelictids really weird and really different from other ciliates is that they're never dividing macronuclei. Just to give you a simple example, that if one Loxodes with 2 macronuclei and 1 micronucleus want to be grown to 2 Loxodes, and each of the daughter cell also need to have 2 macronuclei and 1 micronucleus. And generally for other ciliates, both the macronuclei and the micronuclei can divide and double itself, so that won't be a problem. But for the karyorelictids, it's originally 2 macronuclei will literally separate it into 2 future daughter cells, and at first the micronucleus will go through 1 round of mitosis and to be 2 macronuclei, and continuously to undergo an extra round of mitosis and to let one of them literally become a macronucleus and to refill the number of macronucleus, which is pretty amazing. And given that the micronuclei are supposed to be diploid and there are no significant genomic amplification has been detected within the macronuclei in karyorelictids, so Rick hopes that the macronuclei in karyorelictids are supposed to be nearly diploid, which is para-diploid. And also, each protein coding gene in the macronuclei will roughly have 2 copies. And the division figure I just showed is a really simple example. And sometimes some species, the nuclear behavior can be really complex like this. They will just go crazy. So from here we can see that karyorelictids are really interesting, but unfortunately they're poorly studied, and especially on the nuclear architecture part. Previous studies are focusing on the morphology and done by Rakov using staining and the TEM method during the 1950s to 1990s. And there's no released genomic or transcriptome data. And for the whole group of ciliates, like karyorelictids, around 170 species, there are only 8 proteins on the gene bank, and most of them are Loxodes. So in order to explore more about the nuclear architecture of karyorelictids, we start our project. And just to remember that the hypothesis is protein-coding genes in para-diploid Karyorelictea have low and equal copy numbers as compared to small subunit ribosome gene. And to draw an expected graph, the y-axis will be copy number per cell, and then the x-axis will be different protein-coding genes versus SSU or DNA gene. And what we expect is like this, that all protein-coding genes will roughly have 2 copies, and we have a lot of SSU. And how can we get there? We're mainly using 2 methods. One is the whole genome amplification, which we send our pilot genome sequences and to design our primers. Also, we are analyzing the genomic data, and also allow us to characterize the single-cell qPCR. And the other main method is just the qPCR to help us assess the copy number. I will go a little bit detail on the methods. So for the single-cell whole genomic amplification, literally we pick single Loxodes and put it into a tube, lysate and amplify the genomic DNA using this QIAGEN kit. And we send it to sequence. And from our phylogenomic sequencing, we get 196 million reads and throw out 0.29% of bacterial contamination. And with Xeris, we did preliminary assembly and gene prediction. And from there, I designed Loxodes specific primers for qPCR. And with the qPCR method, we assess the relative copy number. Actually, we're using the standard curve to assess our primers, but because of the whole genome amplification step, we cannot calculate the absolute copy number. We can only, instead, we can only assess the relative copy number. Our goal is to characterize 7 genes from 10 cells, and so far we have assessed 3 genes from 2 cells, which I'm showing here. The 3 genes are SSU, actin, and EF1 alpha. So just to remember that we expect a lot of SSU and only 2 copies roughly for the protein coding genes. And for the next actual observed graph, we specially set EF1 alpha to 1 to estimate relative copy number. So here's our expected graph. And for the actually observed graph, the y-axis is relative copy number, and we set it to log scale. So the EF1 alpha here, we set it to be 1. And for both of the 2 cells, we observe that for each copy of EF1 alpha, there are millions more copies of SSU, which is like we expect. But surprisingly, we get thousands more copies of actins, and I replicate this experiment and got the same answer, which suggests that the differential amplification of protein coding genes might happen. And also, we're testing more genes on more cells and also designing primers for two proteins coding genes from same contig as further control for whole genome amplification. The idea is if it does differential amplification, it will amplify the whole chromosome. And here's a brief conclusion, which I showed before. And from this project, we can see that single-cell genomic approaches can yield insights into an uncultivable clade of ciliates. And also, differential amplification of protein coding genes in Karyorelictea suggests that our model for this class as simple might not be accurate. And also expands on our understanding of genome evolution in ciliates and eukaryotes in general. And at last, I would like to thank Professor Laura Katz and Zaras Anna Olia Puling, who is also here and will present poster during this conference, and the whole CAS lab. And thank you for your attention.
Is it known if this species has tiny chromosomes or has genes organized in longer chromosomes in the macronucleus?
For Karyorelictids?
Yes, or this genus in particular.
I haven't had this kind of research.
Because that would help it amplify differentially individual genes if they're not linked. Do we have other questions? Yes.
You may have mentioned this, but from your assemblies, what is the genome size?
I mean, the genome size?
Yeah. Have you tried getting estimates of genome size? Or I don't know if they exist for karyorelictids, but.
We're still analyzing it, but I think I cannot answer your question now, but maybe later. Thank you.
Any other questions? If not, I want to thank both this speaker and all of the talks this evening, and point out that I think the phylogenetic diversity you've seen just this evening dwarfs that of all the other model systems combined. Thank you very much.